Okay, in this video we're going to introduce the concept of regulation of gene expression in eukaryotes, more specifically in multicellular organisms. Now remember, gene expression is defined as taking the information within your genome, so your genotype, transcribing that into RNA, and translating that RNA into protein, and that protein folding in such a way that it forms hormones or transporters or various structural molecules or enzymes, and all of those give rise to the traits of your cells and therefore the traits of you yourself. And so gene expression is this whole process then of transcription into RNA and then the translation of that messenger RNA into proteins. Now, one of the best ways to understand the importance of the regulation of gene expression is to look at how necessary it is for development. Okay, so what is development? Development is the branch of biology that deals with understanding how a single-celled embryo, a zygote, right? All of us started as a single-cell embryo. You've got your egg, you've got your sperm. That sperm fertilizes the egg, and that first diploid cell that's created is called the zygote. And so that zygote then divides and divides and divides and divides, right? And that one cell gives rise to all of the cells that make up you and me. All of those different cell types as well. Red blood cells, rods and cones of the eye, white blood cells, okay? So this one cell, right, this zygote, has the ability to then give rise to all of the about 270 different cell types within our bodies. One interesting thing here, although this cell, our zygote, can become all of these different specialized cell types, there's really very little evidence and very little examples, almost none, of something called transdifferentiation, where one specialized cell becomes and turns into another specialized cell. Now, when I say specialized cells, another word I can use is differentiated. Okay, this is the form the cells take that they are necessary. The function and structure of that cell is necessary to carry out whatever jobs it needs to do within the body. Now, the question becomes, what is development? What's happening in development? What's happening as this zygote is dividing and dividing and dividing? Is that all it is? It's just mitosis occurring over and over and over and over again with this cell dividing and dividing and dividing. Well, if that was all that was happening, each one of us would be nothing more than a big blob made out of cells that looked like this. It's obviously not the case. We have red blood cells, we have rods and cones, we have macrophages. So as this cell is dividing and as its descendants are dividing, something else is also happening. They are starting to make different proteins necessary to have different structures and different functions. So I've got this question here. What are some differences between these cells? What causes those differences? That's the proteins that they make. Erythrocytes make hemoglobin. All right? That's why they can do what they do. Macrophages do not make hemoglobin. That's why they can't transport oxygen. So different cell types make different proteins, and those proteins are what's going to determine the structure and the function of those different cells. Which means that different genes are being turned on in different cell types, right? Genes are what encode proteins. So if this is making the erythrocyte protein, it means that it is transcribing the hemoglobin protein. It means it is transcribing the hemoglobin gene. This is called differential gene expression. Different cell types make different or express different subtypes of genes, okay? Differential gene expression. Now, there are two ways in which differential gene expression can occur. Let's think about it. One way is that as cells become more and more specialized, they simply lose the DNA that they don't need. A macrophage doesn't need the hemoglobin gene because it's not a red blood cell. And so one hypothesis is as that macrophage develops, as it becomes more and more specialized, it deletes the hemoglobin gene. It deletes all of the genes within its genome that it doesn't need and keeps only the genes necessary to be a macrophage. Right? So that's one possibility, this process of cells deleting DNA that they no longer need because they are a specific cell type. Another possibility is that as the various cell types begin to specialize, they simply turn off genes that they no longer need, which means those genes are still there, which means that the hemoglobin is still in the DNA of this macrophage. It's simply being turned off. It's not being expressed. So those are two possible explanations for differential gene expression. One is that unnecessary genes and cell types are being deleted, and one in which unnecessary genes within cells are being turned off. All right, so now I have another important question. What predictions can we make regarding those possibilities? Let's look at the most important possibility, the possibility that it is actually true. And that is the assumption here that the genes still reside within, all of the genes still reside within each of these specialized cell types. It's just that they're being turned off. Okay, so what that means is the potential for one cell type to become another is there because the genes are there. But we don't see that. We don't see that. So I don't know. Maybe they do really delete all their genes because we can't turn this into this. We can't turn a macrophage into rods and cones. So we don't see transdifferentiation. So let's think of this then. If it's true, if all of the DNA inside the erythrocytes or all the DNA in the macrophages contain all of the genes to make all of the cells, then we should be able to use any cell's DNA to direct the development of all cell types. Well, that's where this little sheep comes into play here. This, if you don't know, I guess, let's see, it was happening in 1989, quite a while ago, and so you guys are younger and younger and younger every year. This is Dolly the sheep. And Dolly was the first example of cloning. And now if you look at the research paper, they said nothing about cloning because the important aspect of this experiment is it demonstrated that if you take a look at any of these specialized cells and take that DNA is capable of giving rise to an entire organism, which means that DNA contains all genes, which means that the model for differential gene expression is a model of regulation, not deletion. So how did this work? Well, what was done, I should say, is that the mother, the surrogate mother, we took an egg from her, so she's the egg donor, and we enucleated it. That means we got rid of the nucleus. So that egg only contains all of the cytoplasm, okay? Doesn't contain the nucleus, which means it doesn't contain the DNA. Then our specialized cell type that we used here were the mammary cells, okay, the mammary gland cells. That was a specialized cell. It's going to be the equivalent of one of these specialized cell types here, where we're not sure whether it has all the DNA or it deleted the unimportant genes. We took a nucleus from one of those mammary cell glands and we put it into this enucleated egg. So now it's that same DNA from the mammary cells, but it's in a different environment. We then implanted that embryo into the surrogate mother, and lo and behold, out popped Dolly. Now, the genetic donor was a Finn Dorset. The egg donor was a Scottish blackface, as was the surrogate mother. And what came out of it? Finn Dorset. Did not have the black face. And then on top of that, when we sequenced the DNA of Dolly, 100% identical to this DNA donor. So what that means is the DNA within these within the nucleus of these specialized cells did have all the genes necessary to give rise to every single cell type and therefore an entire organism. Which means as cells specialize, they don't lose DNA, they simply turn certain genes off and turn certain genes on. But wait a minute, didn't I just say that one cell type can't become another cell type, right? We don't see that. And yet, we saw a mammary cell become everything. Okay, so that's what I'm asking here. But the difference was, and the important thing is that what we did here is we took the DNA from these mammary cells, we took that nucleus and we put it in a new background. Remember, this cytoplasm here from this egg, that's important cytoplasm. There's all kinds of stuff in that cytoplasm that might be influencing the DNA in that nucleus, right? We changed in this cell in transdifferentiation here. We're assuming that that DNA is within the context of still that same cell. But this cloning experiment, we pulled that DNA out of the specialized cell. We put it in a new background that's basically being immersed with all of this cytoplasm and all the various factors that are in it. So there was a guy a little while ago who thought, if the reason that we were able to get a specialized cell's DNA to turn into everything was by putting it into an embryo, maybe we can figure out what it was about that egg. What were those reprogramming factors? And if we can figure out what those reprogramming factors is, then we can actually take those reprogramming factors and instead of putting the nucleus into an egg, we can put the reprogramming factors of the egg into the specialized cell. And that is exactly what this researcher did. Dr. Yamanaka, he won the Nobel Prize for it. He figured that there were 3 important programming factors encoded by 3 different genes. And what he did was he took normal specialized cells from a person like skin cells or fibroblast cells, and instead of putting their nucleus into an embryo, he put the reprogramming factors from an egg into the specialized cells. And sure enough, what happened? They became something called pluripotent stem cells. They sort of became little embryos. And that depending on what sort of solution he incubated these little pluripotent stem cells in, they could become blood cells, they can become gut cells, they can become cardiac muscle cells. And so he called these induced pluripotent stem cells. He took, and by adding these 3 reprogramming genes into these specialized cells, he was able to essentially despecialize them and then was able to re-specialize them and differentiate into other cell types. And again, so important, he won the Nobel Prize for this. Now, what this tells us then is that as cells become specialized, as they differentiate, it becomes very important that they're turning on certain genes and turning off certain genes, right? They don't delete it. We've just spent all those experiments figuring that out. So, regulation is exceedingly important. Spatial regulation, genes must be expressed in the right place. Temporal regulation, genes must be expressed at the right time during development. And dosage regulation, genes must be expressed in proper quantities. Here we have an example of both dosage and spatial regulation where we're looking at the expression of a gene called CROX20 in zebrafish embryos. And there's thousands upon thousands of cells here, and then in the rhombomer, there's rhombomeres 1, 2, 3, 4, 5, and then more rhombomeres down the back here. This is the hindbrain. Notice Crox 20 is only in rhombomere 5 and only in rhombomer 3. No other cells of the embryo. Look how specific that is. Temporal regulation. Again, we're looking at zebrafish embryos, and we're looking at the expression of a gene called RH2. And in stage 1, it's expressed in just this little patch in the eye. And then as time goes on, its expression is increased, increased, and increased until it takes up almost the entire eye. So that's an example of temporal regulation. Depending upon what time in development we look at, we see different expression of a particular gene. Now, if one was to completely mess up, if one was to make a mistake, and a gene was expressed where it isn't supposed to be expressed, so misregulation, we have some really neat examples of this. Here we have a wild-type fruit fly. We've got the eyes, and then we've got the antennae here. Now there's a gene called eyeless, and that eyeless gene is responsible for turning on all the genes necessary for making an eye. Well, normally, obviously, it's expressed in the cells that will become an eye. But if you change where that gene is being expressed, we don't mutate the gene itself at all, just where it's being expressed. If you force it to be expressed in cells that would normally be the antenna, look what happens. Now we have eye tissue on the antenna. There's another gene called antennapedia, which normally gives rise to turning cells into cells of the leg. But again, if you force this gene, you're not mutating the gene at all. The protein is 100% identical to the wild-type protein. You're simply expressing the gene and therefore making the protein in a different set of cells, in the cells that would become antennae. And look at this. You now have legs coming out of the head. So this is a pretty important example of why it's so important that genes are properly regulated. All right. With that said, with the understanding that it's absolutely essential for proper regulation of gene expression, we are going to start the next video with regulating gene expression by regulating the structure of chromatin, the structure of DNA wrapped around these histone core here.